Hello ERGA community, my name is Aureliano Bombarely and I'm here on behalf of the Annotation Committee to give you a summary of our activities during 2021. First of all, let me summarize the committee structure that we have right now. The committee is divided into two groups. One small group composed by 5 to 8 people with different expertises in the field of genome annotation, such as annotation on non-model organisms or transposons. This group is responsible for organizing activities, coordinates these activities with other committees, and discuss them with the ERGA chairs and co-chairs. Then we have a bigger group composed by members of the ERGA community with some interest in genome annotation, from students that want to learn about it to leading experts. We have several communication channels, such as a group email list, a KBase channel, and online meetings that we will start to organize this year, one per month. We have defined 6 functions for our committee. First, to identify the needs of our community in terms of genome annotations. Second, to identify the resources that we can use. Third, to develop recommendations for genome annotations. Fourth, to promote the scientific exchange with seminars, workshops, and discussion tables. Fifth, to enhance the ERGA network for annotation and integrate with other sections. And sixth, to build a framework for the interaction between the partners of the Horizon Europe ERGA project and the ERGA community in the context of genome annotations. We will use several formats, as I mentioned before, like meetings, seminars, and discussion tables. We also organize working groups when it's needed. What is the thing that you should expect from us in 2022? This is our plans. First, we want to hear about your opinions, so feel free to contact us. Second, we are interacting with the pilot committee in 2 different ways, giving some orientations, for example, in the RNA-seq data or isocyclic that we need, and also to organize a discussion tables on January 24 with the sequencing facility involved in the pilot project. Third, we will reinforce our interaction with other committees like the IT committee or the TKT committee. And fourth, we are preparing a series of seminars and discussion tables about different annotation topics such as gene naming and file formats and repeat. Feel free to propose us the topic that you are interested in. So remember, if you contact us, you have all these different channels. Thank you very much for your attention.